Good morning. This is our last day. I hope you are happy. I hope you are not that much tired. We are lucky with the weather. I mean, we had rains, but not that much. Let me introduce our first speaker, Urmas Kõljalg. He is one of my supervisors and the person with the mycological background and the leader of the Unite community. Well, why we're doing this lecture? Because the view from the person from mycology is, in my opinion, like very refreshing for our community. So please, Urmas, you have a floor. Oh, you don't need.
Can you hear me? Thank you, Irina, for the nice introduction. And thank you for inviting me to give this talk on Unite. I also hope that you had nice days and actually it seems to me that you have very nice small global community and you are collaborating, and this is very nice to see. So basically Unite is also a global community and we are working with we started to work with fungal ribosomal DNA ITS data sets. And at first, they came mostly from specimens lodged in collections, also from living specimens. But today we accept ITS sequences from any samples or biosamples as today defined by major data repositories like gene banks and also GBIF. So basically specimen is also specific case of biosample for them. All right. And just to introduce Unite few milestones, and then I show and explain few milestones in more detail. We started more than 20 years ago when a small group of people, taxonomists, ecologists, and bioinformaticians came together in Lund, and we adopted ITS as a standard marker. At that time, of course, there was no word DNA barcoding, so we were not using this term, and even in essence, what we started to do is DNA barcoding. And a few years later, we opened first online key for, at that time, mostly for ectomycorrhizal or mycorrhizal fungi based on ITS sequences. And then a few years later, I think this was very important that Unite and data management platform called PlutoF were split. So PlutoF has its own project and developing data management online platform for any taxon, for any traits and features, so and for any kind of taxon occurrences, including biosamples. But Unite community, we are still using PlutoF as a software to manage our reference data sets and databases. I think very important step for us was 2007 when we started to implement and to include all publicly available at that time fungal ITS sequences. And soon we, of course, implemented all publicly available high-quality ITS sequences from eukaryota. This was a very important step for us. Oh. And soon we also realized that we can't, but it was from the beginning for taxonomists, it was also clear that we can't rely on taxon names. We can, but in most cases, we were not able to identify or to put full species name on ITS sequences. And of course, this generated many issues for communication. How you build, and you know this very well, how do you build identification key when you don't have species names? So will you use names of genera or families or even higher level because you don't have a more exact name? And then how you communicate. So Boletus, sp, 1, second, third, etc. So we decided that we were working few years, and then we implemented so-called taxon hypothesis paradigm. And species hypothesis is specific case of taxon hypothesis. And then we also started to generate persistent identifiers for those species through different versions we were computing. And all these persistent identifiers, they are connected to taxonomic backbone. So, and then this identifier can be connected to the full species name, if there is full species name available, but it can be connected in backbone also on genus level or higher level. In this way, everybody who were using our resources, Unite resources, they could identify and to use in parallel taxon name as well as persistent identifier, which is unique and stable in space and time. And 2013, we started to use DOIs. So you know very well DOIs, which are used also for research papers. But there is also a specific community called DataCite. And this community actually providing their members option to use the same DOIs for research data sets. And so this is also why we are using PlutoF, because PlutoF is member of this data site and can publish unlimited number, millions and millions of DOIs. And we did this. So every species hypothesis, or better to say, every taxon hypothesis has unique DOI, and all DOIs through different versions are connected. All right, this was very important. And this is current view of single species hypothesis in PlutoF, how we work. So this is, of course, special case when you have wide screen monitor. So then you can have, as long as your monitor is alignment of the species, and so you can work, and then it's easy to see places where your species has variation or potential mistakes, and etc. Because all these alignments are computed automatically based on methodology implemented in Unite. But I will show you, if I find here laser. Yes, I will show you this part of the species hypothesis. And here you can see that I was telling you about communication of species, that every species hypothesis has DOI, this one here, and DOI must have separate home base. This is the rule. And of course, there is specific page for DOI where you can go and download this data set behind of DOI. Basically, you can go and download alignment, all sequences, all metadata of this particular species hypothesis. And it is always connected to taxonomic backbone. So it must be connected to the taxon name. If it is on species level, that's very fine, but and very good, excellent. But often we don't have, because we have many undescribed species in the system. But I will come to this back later. How much time I have? Okay, thank you. But also you see here are links for the kingdom level taxon hypothesis. So basically it means you can click here, go to homepage of DOI for taxon hypothesis for fungi and download everything into your computer and start to work whatever you want to do with data. Of course, I will show you later, you can download everything from Unite because it is free source and start your own studies, in-house studies and analysis. All right. I have to be closer, it seems to be. And then a few years ago, GBAF decided, because they saw that there are more and more environmental DNA data and studies, and GBIF wanted them inside their database, data portal. But the issue was that, as I told you, most eDNA studies and unknowns, they don't have full species name, and they are very difficult to connect to taxonomic backbone, and there was no common rules how to actually submit those taxon occurrences from eDNA studies to GBAF. And then they realized that Unite system, where we have DOIs for every species connected to taxonomic backbone, which is very much and very similar to GBAF, because partly Pluto-F is using GBAF taxonomic backbone, or better to say catalog of life. So they implemented Unite DOIs into GBIF taxonomic backbone. So every DOI went to their place. And this was big change, I would say, for eDNA data submission to public data portals. And then, of course, we incorporated also long ITS sequences from backbio environmental studies, because so far we had mostly Sanger sequences from single specimens or single, let's say, living cultures or some other biosamples. And then we decided also, because eDNA studies became very important users of Unite system, and we were asked to cover all eukaryota, because you can imagine when you took your biosample from environment and you extract all your DNA in this sample, and you sequence, let's say, ITS and part of SSU and LSU, you want to identify everything, all unknown sequences, as a single run, basically, at first at least. And even if you are interested about fungi, how do you know that this sequence represent fungi? You need sequences from Animalia, from Viridiplanta, etc., from protists. So we covered 4 years ago eukaryota for this purpose, but it's developing further now. And there are people who are very actively actually annotating plant ITS sequences in Unite, forming their own database. And I think very important step was last year when, better to say Pluto-F, but this was thanks to Unite. Pluto-F started a collaboration, very close collaboration with European Nucleotide Archive and GBIF. And then there was, so I will focus now a few slides on these two topics. This slide is from January this year, very common, and I think many of you know the species Boletus edulis, yes? Very common and beautiful edible fungus you probably saw yesterday. And if you look this taxonomy part of Boletus edulis, of course, a lot of synonyms. And in the end, you will see immediate children unranked, and then you see short DOIs from UNITE. So this is how it looks in GBIF when you look for the species. And sometimes, of course, they are under the genus name, under the family name, or if they are identified on kingdom level, so they are immediate children under the kingdom name. But this will give a possibility for researchers to upload their taxonomy occurrences from eDNA studies if they use ITS and those DOIs from UNITE datasets. All right, but you see there are three species hypotheses under the Boletus edulis. And then there are two options, actually. Either this Boletus edulis includes more species than just one, or there is mistake. And how we work, I will show what happened in January. So I was searching immediately in BlueTF for those Boletus edulis species hypotheses. And I found three of them as marked in GBIF taxonomic backbone. And one of those is this, we can think of real Boletus edulis with more than 1,000 sequences inside this species hypothesis in UNITE. But then there are two which include only one sequence. And one comes from, I don't remember, one comes from China, another from Nigeria. So, and why they are called Boletus edulis? Because those two sequences, they were submitted to GenBank in US NCBI database as Boletus edulis sequences from China, Nigeria and China. And so they started to fly around as Boletus edulis representative sequences. How you can comment, yes? Yes, so this is, and what UNITE do in such cases? We have system where you can annotate every single sequence, not in UNITE, but also in BlueTF, of course. If you have your own database in BlueTF, you can annotate every sequence you have. And then you can actually add new identifications. So, and we did for this two Boletus edulis species hypothesis, and we identified them as Boletus. And now this project called Bicycle, this is European Community Project for data, biodiversity data infrastructures, including European Nucleotide Archive, GBIF, but also BlueTF and Elixir, which is infrastructure for biological data, better to say molecular data in Europe. And so these three infrastructures, they build connection between them and now unite those sequence annotations can be sent to ENA immediately. When you annotate sequence in, let's say we annotate in UNITE databases and we click button, send to ENA, because BlueTF is official broker for ENA and it goes. and UNITE is trusted database for them. And there is then, for this sequence, which is, I think it was from Chinese, yes, Chinese sequence of Boletus edulis. You see name is here. It is originally, I think it was submitted to American Gene Bank, but they share data. And then there is a menu called third-party curations. Not annotations, but curations, because annotations in genetics has different meaning, a little bit different about genomes. You are annotating genomes. All right. And then there is data that it is identified as Boletus because we don't have correct name. The same with Nigerian sequence. And then yesterday I went back to GBIF because the same data are, I don't know how often UNITE sends to GBIF, but it goes, I think, quite often. So, and now you see Boletus edulis has only one immediate children because those two others, they are now under the Boletus. We don't know. So if you go to Gene Bank, there is still, and you identify your Nigerian sequence, and the closest match is Nigerian, you will get name Boletus edulis, and they have just link out to UNITE. So if you're not smart enough to link out to UNITE or to go to GBIF to see what they think about sequence, then you use edulis and this. And then there are more and more probably wrongly identified sequences will be deposited. All right, you know very well the issue with myxomycetes, I'm sure. Okay. And when you click on this species hypothesis in GBIF, you will see more data. And they now show how many taxon occurrences, and they all are coming, in this case, I know, from soil samples. And there are 2 studies who actually uploaded those occurrences from very different soil samples globally. Yes, so they were submitted as with DOI, and then the taxon occurrence found correct place in the taxonomic backbone. And it's interesting to see that there was one study using hypothesis, current hypothesis 9, and another study was using version 7. But you see here, they also show that those earlier versions, actually they are synonyms for this current version. And even more, when you go to UNITE, you see that all individual sequences are connected through versions. So you actually can run automated and reanalyze your data very easily because individuals are always connected through those DOIs. All right. And if you have your unknown sequences from any biosamples, and you want to publish them in GBIF, then you can use in BlueTooth matching analysis. This software is also available in GitHub, so you can download and actually set up your in-house analysis. And then you will get data in the format you can go and submit to GBIF also through BlueTooth automatically. So it is very simple in the end, but you have to learn how to use those resources. But we have public online resources page, specific page in our homepage, where we have our data sets freely available in different formats. You click on Chime here, and then you will get a lot of different reference data sets in format which can feed directly to Chime pipeline for analysis. So, and people do this. So last year, there were almost 8,000 downloads of reference data sets. And these are 5 most common reasons, because now we are asking people when they download, because everything is free. Only thing we ask, what is the purpose? And they are happy to give this. So we have some kind of, and they can give other feedback as well. So we know how to develop Unite resources further. It's some kind of survey when they download. And they are happy to give, we see most of them happy to give also email when new version is available. So only then they will get a small note that, okay, now there is new version. So if you want to run your in-house analysis, please come and upgrade your reference data set. So current version we have, when we force 1.5% difference between 2 sister species, we will get almost 300,000 fungal species hypothesis. When we force smaller distance between 2 sister species, 1% difference, then we will get, you see, 550,000 fungal species hypothesis in current version. Probably as a taxonomist, you know very well how it works. So it depends on your opinion on species, what kind of concept you have or definition for your species. And ecologists, when they analyze their data, they mostly focus on intraspecific variation. They force that, okay, now we have, let's say, 2% or 3% intraspecific variation, and then next species will start. But in Unite, from the beginning of species or taxon hypothesis paradigm, we are working mostly with distance between species, and we force gap between species on different levels. So if you are familiar with species hypothesis, you see that there are different colors showing different levels, distances. So we actually calculate 0.5% difference, 1%, 1.5, 2, etc. So today we recommend to use mostly 1% distance between sister species. But then you have much more species, of course. But still, this is very conservative, at least in mycology, because, you know, ITS is still quite conservative. So, and of course, as a taxonomist, and maybe you as well, I would like to have a gene marker which can identify individuals, or even better, different cells of the same individual. But there is no such marker as far as we know today, unfortunately. So ITS is quite conservative, and this is still conservative. If you look for ecologists, and they use this intraspecific variation to calculate their OTUs, they would have, for the same data, I would say 5 million or around species, if you force this intraspecific variation. But this means that we have some species which have quite wide intraspecific variation, more than 4, 5, even 6%. All right. So, and then of course we have question, is it still species? All right, and other eukaryotes. So we have, if you use 1.5%, so we have more than 60,000 plant species hypothesis based on ITS, and 1%, then we have 95,000. Not so much animals, because mostly CO1 is used for animals. But some groups, they are using also ITS, and then there are ITS sequences available in public data repositories. All right, other groups, and I will show Amöbozoa on different slides because this is the yes. Am I right? This is the group you are studying. No? Okay. Yes, of course, names can be different because different classifications have different names. And this is Amoebozoa. So it has not so many species hypotheses in our system, at least for 1.5%. It's just a little bit over 700. But the interesting is that 144 are unidentified. So they are identified on Amoebozoa level. We don't know where they belong. So yeah, I have no idea, of course, because I'm studying fungi by myself. So, but often in case of fungi, if we see those unidentified, either they are mistakes or often they represent new lineage undescribed, very high level. So they are most interesting to study, actually. Not most interesting, but they are interesting for taxonomy. All right. All right, and this is now Myxocastria or Myxocastria in some other taxonomic backbones or Myxomycetes. Yes, you see not very many, just 26 species hypothesis for Myxocastria. But it's interesting to see that many of those sequences are now coming also from soil samples. And this is one species hypothesis from Physarum, and all sequences are coming from soil samples from Estonia in this case, because Estonia has been very well studied for soil based on back biosequences, and we accept only full ITS sequences. So all Illumina studies or previous studies with ITS1 or ITS2, they will not end up in this species hypothesis. You can identify your, let's say, ITS2 against unite resources, but we don't accept short because it will make many taxa. Yeah, there will be a lot of mistakes based on our calculation computing methodology. So we don't accept for species computing those short sequences. All right. So obviously there are much more. Yes, so it's interesting to see that they are coming, but not so often. I don't know the reason, you know, probably you can tell. Okay, I will not show maybe this so much, but it shows that most of the species and sequences we have in our system is still from fungi. And of course, fungi is the probably most diverse taxon when you think about soil examples, together with, of course, also insects and groups, animal groups. All right. I think I've been using most of my time. Okay, this is here. I was comparing for another talk. Do you know MetaPR2 database? It's mostly for protist identification and based on small subunit sequences. Okay, it seems to be you don't know, but this is one, you know. Okay, have you been using it? Oh, okay, good. Yeah, I met them this summer, so we were talking about collaboration. So to connect somehow those reference databases so that it's more easy for users to switch between two different markers. But the interesting thing here is that because they are focusing on protists only, that the most, let's say, species-rich groups in their reference data set are the same as in ITS-based UNITE data sets. For, of course, we have here unclassified. So this means unclassified on kingdom level. Many are, of course, just mistakes. Some kind can be chimeras, etc. But there might be, of course, inside very interesting taxa. I don't want to say new kingdom level, but possible, especially when we think about protists. But this was very interesting when we shared SSU and ITS data, and we saw that the number, abundance of species in different kingdom level inside protists in UNITE and PR2 database are actually overlapping nicely. Yes, current version has over 8 million ITS sequences in UNITE, and you see that only fraction is now coming from Sanger sequencing. Mostly they are coming from high throughput sequencing studies, and I think almost probably 99% of those are from PacBio sequencing efforts. All right, because then we cover also those taxa which are not easily collected from the field as specimens, as sporocarps of the fungi. And then when we think about, and this was about sequences, now about species hypothesis, and you see that those two, let's say, data sets of PacBio sequences and Sanger sequences, they stay separately, and only 3% of species hypotheses in UNITE include both Sanger and PacBio sequences from environment, which is also interesting to see. But this makes UNITE very good resource because it includes high quality PacBio sequences, so you can actually identify many unknown taxa, including probably undescribed taxa. And UNITE, how it works. So we started as a very relaxed community, and we just came together, we discussed things. There were no real rules. There were very democratic discussions and sometimes heated discussions, but always we found some consensus and we developed. But a few years ago, it was quite clear that when we started close collaboration with European Nucleotide Archive, with GBIF, then it was clear that we are nobody because we have no legal structure. So somehow we were pushed, and you know, researchers, we don't want to do very much bureaucracy, but we were pushed to do. So we started a non-profit association, global association, which is now almost 2 years old. And we gradually changed the governance of UNITE because we need to have body to lead, and we are growing. We are inviting different people who have been providing data and working and proposing ideas. We are inviting them to become official members of this association. But this is also some bureaucracy. Every year we have to meet and to do some formal work to fill papers and reports for the authorities to show what we did. But this now we are able to have a real contracts with GBIF. We had already with GBIF contract for collaboration. And most importantly, now we can be partners or apply by ourselves projects from European Commission, because this association is registered by European Commission, and we can actually apply. And that's very good, because before we were just using our own research money for sequencing and for computing. All right. And why I'm showing this is if you are interested somehow to build up your own myxomycetes group working with UNITE, then you are welcome, of course. All right. And BlueTef is very important for UNITE, and BlueTef is developed here in our university as well, and Kessi Abarenko is very good link, because she's my former PhD student. She's informatician, but she got also education in biology. So today she's mostly working with software, and she's the person expert for computing taxonomy hypothesis. So this is her duty, and she's very good to translate our biologists or taxonomists' wishes to those IT people shown in this slide who are developing BlueTef. So far they accept us as a major developing force for BlueTef. But BlueTef is growing and covering other data fields as well. So we will see how it works in future. I hope it will be okay. And that's all. Thank you. Okay, sorry. Here was also okay. In the end. Okay.
Yeah, thanks for this very, very interesting talk, giving us insights how such a database can work and how such an enterprise develops from tiny beginnings to a real huge organization. We are a little bit sad that we can't be so much part of the community right now, since in myxomycetes, and that's the thing we share with other groups of protists, barcoding seems mainly to focus on the 18S rDNA sequences, which we found to be quite useful, whereas the handful of studies with ITS sequences found that the variation is so large that it's difficult to build up species hypotheses with it. And my question would be, when we really want to build up a database with our sequences, which are still right now mostly SSU sequences, I would think the whole community has at least a couple of thousand, huh? Close to 10,000, I think we would be close to 10,000 SSU sequences right now. What would be the right database from your point of view to just match with?
I have to, because I was not talking about outlook of the UNITE, but currently we are actually including all marker genes, or let's say DNA barcoding marker genes sensulato into UNITE. We are not sure we will start to compute species hypotheses, but we want them to be linked, and at least to calculate taxon hypothesis, because for us SSU is above ITS-based species. Usually we see that if we calculate those taxon hypotheses, or let's say 3%, we force 3% distance, we see that we have groups of species together. And often because we have been analyzing SSU for a long time now, because we wanted to know how many of our species are inside one SSU-based species or taxon. And what we saw, at least for fungi, that usually we have several species hypotheses inside one SSU-based species, or whatever on what level you call it. So you can use, and you can have a help from Unite and to use all these resources. So of course, there are specific, as I showed you, specific SSU-based databases. There are several actually. And I can tell you also that if you come to Unite to use Unite resources, or Bluetooth, better to say, because you don't need to show your data to Unite. You can have your own database in Bluetooth, but they are linked immediately if you want, because they use the same data fields. And you can start to use the same, actually, a set of features, annotating sequences, adding metadata, combining eDNA, Sanger sequencing data, all this together, and also publish in GBIF, and also publish in European Nucleotide Archive. So this is more or less automated, and you can download automatically from GenBank's data as well, and from GBIF, obviously. So this is one possibility, but I'm talking about from my point of view. So you have to make your decision by yourself. But as I said, we are working, and this autumn we will have full database of all publicly available of around, I think, 10 different barcoding marker genes. How, I can't tell you how we will compute, because we don't know, still we are discussing how we will compute and how they are connected. So, because GBIF is interested, they want to have single data portal for biodiversity. So they are pushing us to have solution, and other databases, common solution for species or species hypothesis. So they are pushing us to have specific project which Unite will lead together with other data portals and repositories. So to say, to come together and to have a single solution for users, but for us, there are different databases and different communities working. But the solution for users and GBIF is single, the same. Not the single, but how to say, I don't know.
Uniform.
Yes, so that those who are using GBIF, they see single data base, so to say, global. They can use, they can search, they can download. But citations go to different databases, to different people. And we are working on that as well. Because right now, you know, you go to GBIF, you download data, and you will get DOI, you have to cite in paper. And they are citing then those who were actually uploading data to GBIF, not the owner, let's say, of who was collecting specimen, who was sequencing this specimen. They don't get direct citations. But we are working right now so that all those data will be atomized, even inside one single specimen data collector, who identified, who sequenced, they all will get citations. And this comes together as a single system, but it takes few years before it is up and running. If this is okay, answer. There was another question. Yes, you just, young man, you have loud voice, probably.
Yeah, Urmas, my name is Dmitry Leontiev. It is a pleasure to meet you at last. I have two questions, one small and one bigger. Small one is, are you planning, or you already have this, to connect the individual sequence to the original sequencer chromatogram file, the EB1 file? Because sometimes everybody knows that sometimes you see the individual difference, the single letter different, your sequence from something from GenBank, and you cannot check who is right, because you cannot check peaks in the GenBank chromatogram.
Yes, you can do this in Bluetooth. We don't do this as a Unite, as a community. but those who are working with smaller groups, they can upload. So every specimen, every sequence, because one specimen can have hundreds of sequences, every sequence can have a chromatogram, so you can check. So that's no issue, but we don't do for our sequences because it's too much for us. But our people, they are working with one genus or group of species. Of course, they are doing. But I don't know how many chromatograms there are in Pluto F behind of sequences. But you can do this. You download GenBank sequences, and if you have chromatogram for the specific sequence, you can upload to Pluto F and work. So, and you can actually see online those chromatograms. So this is not the issue.
Great, thank you. So this is possible.
Yeah.
And the second question, if I understand well, the GBIF wants to make a catalog of everything, so they need names. And if they don't have names, then they provide some kind of individual codes for OTUs and so on. But taxonomists know that genetic distances between species are very different, and they're also different depending on the marker gene you use. So do you really expect that this formal separation based on the uniform genetic distance, uniform barcode gap, will really work for such a huge groups as fungi, for example?
No, it is not working. So UNITE is not, how to say, doing basic taxonomy. No, we are not doing. We are trying to help to identify and communicate, most importantly, communicate those where we don't have good taxon name on species or even genus level. This is the idea, so that still we can collect those data as a biodiversity taxon occurrences, analyze them together, and address specific questions. Because right now, if every ecological or eDNA study, they have their own OTU numbers, then next studies must start from the beginning all the time. And that's not possible, and it's very bad for taxonomy. So we always said that we are not doing taxonomy in the sense that we are not describing species. We are just, how to say, we are making groups based on specific rules. And actually, in our system, you can force to change this distance or content of the species. You can remove single individuals and add. But it is very time-consuming, as you know. Taxonomy is very exact science, but very time-consuming. So, but you see, when name of the unknown species present as sequences in UNITE comes 10 or 20 years later, the system is built so that it goes back in time, this name, and will meet the very first version of this species based on sequences, and then it will be in GBIF the same, because all these versions are connected. Of course, it is difficult because sometimes we see that one species hypothesis will fuse with another, because sequence is coming between them, and we see that they come together as a single, or they split also. Because sometimes we have one species, but there are chimeras, not chimeras, but mistakes. And these sequences will be filtered out. And then we see that one species hypothesis will be split. There are two species. And it looks better for taxonomy. But this is not, we are not describing species. You have to, this is why we are using those DOIs. We are not putting names, but DOI is connected always to backbone. So I hope this is clear.
Yeah.
Because I'm taxonomist and I do taxonomy, but it's extremely useful for me and some other taxonomists. They told me that when they start from species hypothesis, and then they have idea what species they should study or where they have undescribed species.
Thank you.
Yes. Go.
Which means, in other words, one of your objects, given a DOI, you never change the DOI, but it will change its taxonomic affiliation with progress in science.
Oh, yes. And individuals are leaving and coming inside, but then the DOI is different version of the previous. But all individuals are in the system. We know from where they came and where they went. But the issue is that name comes to single individual. And follows this. So you can even imagine the situation that in one version there are two species names, or if we have sequences from type material, and one is synonym of another. But then they will be split in future versions. So the synonym name will become the real accepted name as another one. So this may happen also. And that's how we try to build the system.
The problem is the individual researchers have to do the annotations or curations.
Yes, and you can curate closed system seen by you or by your group of people or your students together. They are locked. And many people annotate actually sequences in BlueTF. They are locked as a project. And then after a few years, they push button, open them. And then they are available for GBIF. You may send them by yourself to GBIF, etc. So you don't need to open data in BlueTF. You can lock them. But we want to have in future some kind of moratorium, because we have some issues when very good people, they left us for clear reason. And their data are sometimes, some part of data are locked. What can we do then? And they are very important data, taxonomically, very important data. So we think that we push a moratorium for data, how long you can lock, but you can always prolong it. But you will get email, okay, this 2-year period is over next month. Would you like to lock them further? Then you have to go actively and push button that I want them still to be locked. If you don't do, then, and if person cannot do so, data will fly and available for others. So this is idea, but we don't know, can we do this? Is it some kind of, it's difficult today. Oh, sorry, my answers are too long.
Can I have a... are you planning to work together with the protist ribosomal reference database, the PR2 database that you were talking about?
Yes.
Because as I said, I know one of the curators is Anna Maria Fioradono. So we all, lots of us know her. She has a background in myxomycetes that we are studying.
Yeah.
So
I don't know if she was in Barcelona in the beginning of July. We had meeting. Unite was invited. Also Worms, if you know the database taxon names. And of course, GeneBank was there. But it was organized by PR2 and other databases of SSU. So at least we decided that we will collaborate. And because those data, different market genes must be connected to each other. So I didn't say, but we have now collaboration with Bold for some time. There will be some text coming out, some kind of outlook how to build common system, Bold and Unite, so that users will benefit from that. So there are a few things we are discussing, of course. We have different opinions, but this is also, it's even further this collaboration between Bold and Unite now. But there will be, I'm sure. because they are actually, some of them are studying also Plute, I know, but they don't know, will they develop their own data management platform or not? So, yes. So, thank you. Interesting. Nice meeting.